Good day. This is the Professor, CBMD PhD. And today's Monday, March 20th, 2023. It is 1:17 and 15 seconds, a.m. Eastern Standard Time. And today's research, pancreatitis. So let's hop to it here. Vitamin A may reduce pancreatitis risk during whole treatment. Date March 15, 2023. Source, Cincinnati's Children's Hospital Medical Center. As for people with whole treatment, with the enzyme asparaginase helps starve cancer cells by reducing the amount of asparagine circulating in the blood, which the cancer cells need but cannot make themselves. The medication, often used in combination with other chemotherapies, is given via injection into a vein, muscle, or under the skin. However, an estimated 2% to 10% of asparaginase users develop inflammation of the pancreas in reaction to asparaginase treatment. For a third of these people, the symptoms can be severe. Researchers developed predictive analytics using over 100 million data points, encompassing gene expression data, small molecule data, and electronic health records, to understand more of the mechanisms driving asparaginase-associated pancreatitis, and identify potential interventions to prevent or mitigate asparaginase-associated pancreatitis. As first they analyzed massive amounts of gene expression data to reveal that gene activity associated with asparaginase of pancreatitis might be reversed by retinoids, vitamin A and its analogs. The team found more supporting evidence by mining millions of electronic health records from the TriNetX database and the U.S. Federal Drug Administration Adverse Events Reporting System. And this number crunching and predictive analytics work included the use of Adverse Event Reporting System Mind software. The research team also studied data from mice experiments and compared plasma samples from people with all who developed pancreatitis and those who did not. Ultimately, the team established two sets of human real-world experiences. They found that only 1.4% of patients treated with asparaginase developed pancreatitis when they were also taking vitamin A, in contrast to 3.4% of patients who did not. As concomitant use of vitamin A correlated with a 60% reduction in the risk of asparaginase-associated pancreatitis, lower amounts of dietary vitamin A correlated with increased risk and severity of asparaginase-associated pancreatitis. And this study demonstrates the potential of mining real-world data to identify therapy modifiers for improving patient outcomes in cases where a primary drug induces toxicity but is critical to therapy, such as asparaginase. Therapy modifiers, such as vitamin A and its analogs, may be immediate relevance to patients on asparaginase and at risk of asparaginase-associated pancreatitis. The study highlights the power of heterogeneous data integration and analysis and translational research. By leveraging existing omics and patient-centric data in a systems approach, they were able to identify new insights into the development of asparaginase-associated pancreatitis and potential interventions to prevent or mitigate this side effect. In some ways, learning from this study can be applied immediately to patient care. However, more clinical research is needed to establish how much vitamin A would be needed to protect all patients from pancreatitis, or whether a protective level could be achieved by diet or via supplements. In fact, target vitamin A levels may need to vary according to individual differences in metabolism. And this is a Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.